Hello, dear students. Today we will continue more about the circulatory system. So let's start. First of all, we will be studying about the various blood vessels. So there are three different types of blood vessels: the artery, the capillary, and the vein. The artery is most muscular, has got a narrow lumen, and is a blood vessel which carries blood away from heart. The vein, on the other hand, has got a very thin muscular layer, has got a very wide lumen, and has got these pocket shaped valves throughout its length. The vein carries blood towards the heart from an organ. The capillary is the structure which connects an artery and a vein together. The artery and vein have got three layers: tunica interna, the innermost layer, which is having a single layered endothelium, the basement membrane, and elastic lamina, an elastic layer, the tunica media, which is basically having the smooth muscles. These muscles are not under the control of an individual. And the tunica externa, that is the external layer. So these three layers are found in both the artery and the vein. But in a capillary, it only has a single layered endothelium, so that the exchange of gases, the exchange of nutrients, the exchange of waste material can take place across the single layered epithelium of the capillary. So the valves which are found in the vein, they have a very special function. When the blood is flowing from the organ to the heart, it is flowing against gravity in most of the cases. This blood may try to go back, return back to the organ. When it tries to do so, the valve gets filled with blood, and these pocket shaped valves block the opening of the vein. So the blood cannot go back, it can only go forward. So the valve is basically meant to allow unidirectional flow of blood from the organ to the heart. Now, there are these terms: vasoconstriction and vasodilation. These two are properties of an artery. An artery, under its normal circumstances, will allow a good amount of blood to flow through. But since the artery has a very thick muscular layer, it can dilate itself to increase its diameter, which will be called as vasodilation, or it can constrict itself to reduce the diameter, which is called as vasoconstriction. Vasoconstriction is used when there is blood loss, as in case of bleeding or an injury, to reduce the blood flow to the affected area. Vasodilation is used when the blood flow needs to be increased to a particular area. So, this is what vasoconstriction will do. It will reduce the blood flow to a particular area, and vasodilation will increase the blood flow to the same area as and when needed by the body. Now, there are differences between artery and vein, both in their structure and in their function. So let's study the structural differences. The artery is thick, more muscular, whereas the vein is thin and less muscular. The artery has elastic walls. The vein is non-elastic. It doesn't have elastic walls. The artery is deep-seated. That means it is present below muscular layer, not exposed to the surface. There are some exceptions, but majority of the arteries are present below the muscles. The vein, on the other hand, is superficial. It is present above in the upper layers, just below the skin. The artery has a narrow lumen, a small lumen, whereas the vein has a wide lumen. The artery has no valves. The vein has valves. The artery is progressively branched and it decreases in size. The vein progressively unites and ultimately increases in size. The artery forms arterioles and the vein arises from venules. So these are the structural differences between the artery and a vein. Now we have the functional differences between the artery and a vein. The artery basically is a blood vessel which will carry blood away from heart. The vein will carry blood towards the heart. The artery does not collapse when empty, whereas the vein collapses when empty. In the artery, the blood flows in jerks because as the heart is pumping and pushing the blood in the artery, a jerk will be produced. That jerk will be called as the pulse, which you can feel in the radial artery of your arm. So the blood flows in the artery in jerks, whereas in the vein, the blood flow is very continuous. The blood is coming from the organ, which will not be pumping it. Only heart is the pumping organ, so blood flow is more gentle and more continuous. The artery can constrict or it can dilate. The vein, they cannot constrict. The artery carries oxygenated blood, except for pulmonary artery, whereas all veins, they carry deoxygenated blood except for pulmonary vein. So these are the functional differences between an artery and a vein. Now next, what you're studying is called as the pulse. This is the radial artery. This is an artery which comes very close to the surface of skin, so you can feel the pulse here. The pulse is basically alternate expansion and elastic recoil of the wall of the artery during ventricular systole. Whenever the heart is pumping blood, and especially the ventricles are pushing the blood towards the artery, toward the aorta. To be specific, the aorta will divide and send that blood to the arteries and various organs. In doing so, the same amount of pressure as exerted by the ventricles towards the aorta will be transmitted forward. So the blood flows in jerks. So there is an expansion when the blood is flowing forward and a little recoil afterwards. This is felt as a pulse in the arteries. This is the pulse which is taken by the doctors to measure your heartbeat and hence let you know whether your heartbeat is increasing or is maintaining its normal state. So the pulse is measured by using some pulse points. This is the most common pulse point called as the radial artery pulse point. Now, what is blood pressure? Blood pressure is the pressure with which the blood flows and exerts the pressure on the arterial walls. Now, this pressure is measured in two values, systolic and diastolic. Systolic pressure is when the ventricles are contracting. Diastolic pressure is when the ventricles are relaxing. We measure ventricular systole and ventricular diastole when we are measuring pressure. So the ventricular systole is 100 to 140 millimeter of mercury for a healthy person, and the ventricular diastole for a healthy person is 60 to 80 millimeters of mercury. These two pressures are measured by using an instrument called sphygmomanometer. You must have seen doctors tying this cuff around this belt around arm and keeping a stethoscope here to monitor the blood pressure of a person. So this device is called as a sphygmomanometer. Now, then there is this concept of double circulation. The blood comes to your heart two times. The blood goes from the right ventricle to the lungs for oxygenation, then comes back to the left auricle. So the blood is leaving heart once and coming back again. Now this blood goes from the left ventricle to the entire body systems and comes back again to the right auricle. So your blood leaves the heart twice, comes back to the heart twice. Therefore, this kind of circulation is called as double circulation. Now, if you were to see the position of heart and the various blood vessels, the vena cava carries deoxygenated blood from the entire body, whether it is superior or inferior, into the right auricle. This blood goes to the lungs for oxygenation via the pulmonary artery. Ultimately, from the lungs, the blood comes to the left auricle via the pulmonary veins. There are 4 pulmonary veins and 2 pulmonary arteries. Once this blood has come to the left auricle, it goes to the left ventricle, and via aorta, it is supplied to the entire body. This will go to the liver, to the kidneys, to the stomach, to the intestine, to the legs, to the arms, to the shoulders, to the head, every body part. And this entire blood will get deoxygenated and come back again via vena cava to the heart. The blood circulation which is going to the body systems is called systemic circulation, whereas the blood circulation which is going to the lungs is called as pulmonary circulation. So we have two types of circulation, pulmonary and systemic. Now, in all these, if you notice, one type of blood vessel displays a little difference in behavior. Normally, all blood vessels come after originating from the heart, enter into an organ. So the artery comes to the organ, supplies blood, then leaves as a vein and rejoins the heart supply. Only in case of this particular blood vessel, which is called as the hepatic portal vein, is the blood going from one organ to the other organ. The organs here are stomach and intestines to the liver. None of these organs are heart. So any such vein is called as a portal vein. And since this is supplying to the liver, therefore this is called as the hepatic portal vein. So the hepatic portal system basically takes the blood from the stomach and the intestine. Instead of directly sending it to the vena cava, send it to the liver. The liver will detoxify it, use the nutrients, store the nutrients, and then this blood will be passed via vena cava to the heart. So this is in case of any normal organ. The blood comes from the heart, then the artery will carry that to the organ, divide it into capillaries. The capillaries, after giving oxygen, will form a vein, and that vein goes to the heart. But in case of the hepatic portal system, the blood comes to the stomach and intestine. Once the digestion process is complete, the hepatic portal vein is formed here. And this blood now goes to the liver. In the liver, the nutrients are absorbed or stored, the toxins are detoxified, and then this blood goes to the hepatic vein and back to your heart. This step is of immense importance. If in case somebody has consumed any poisonous substance, the liver detoxifies it. After digestion, whatever nutrients are formed, the liver will be utilizing them. Glucose is stored as glycogen, amino acids are broken down to form urea in the liver. Any toxic substance which enters in your food system, it is detoxified here in the liver. So this step is of immense importance. That is why your body has formed a hepatic portal vein here. A portal vein opens in capillaries and ends in capillaries. Okay, it leaves one organ and enters another organ, and none of these organs is heart. That's the basic requirement for a portal vein. Now we have lymph. Lymph is another straw-colored liquid which is basically found in lymph vessels. It is also found in lymph glands like spleen, like tonsils. So you have certain lymph nodes in the body where the lymph is formed and stored. It is rich in WBCs and helps in maintaining immunity in the body, helps in protecting your body from diseases. The lymph is flowing in these green colored structures, these vessels which you are seeing. These vessels also have valves because lymph has no pumping organ. Now, ultimately, lymph will be moving forward or backward because it is squeezed. These vessels are squeezed inside your muscles. So when you move your muscles, the lymph moves forward or backward. And ultimately, the lymph is poured at this point where the vena cava is entering into the right auricle. So it becomes a part of your blood circulation. Again, the blood plasma and WBCs will ooze out of the blood capillaries and again become a part of the lymph. So in this way, the nutrients and the waste material are also circulated via the lymph. So these are the various lymph nodes which are found in the body and the various lymph glands which are found in the body. So lymph, as I told you earlier itself, is extremely important for providing protection and immunity against a number of diseases. So what are the functions of lymph? It supplies nutrition and oxygen to those parts where blood cannot reach. It drains excess tissue fluid, metabolites, and returns proteins to blood from tissue spaces. So whenever proteins are not able to return from the tissue spaces, the lymph helps in returning those proteins. It helps in fat absorption by lacteals. Now, lacteals are structures found inside the small intestine. They are basically involved in absorption of fatty acid and glycerol. The lymphocytes and monocytes, which are found in lymph, they basically are types of WBCs. They help to defend the body against diseases. They remove bacteria from body tissue by phagocytosis. So these are all the various functions. They localize infection and prevent it from spreading in the entire body. So once the WBCs have located any source of infection, the WBCs accumulate at that point and the infection is localized, contained at a particular area. So you have the next lymphatic organ. In fact, the largest lymphatic organ, that is the spleen. The spleen is present behind the stomach. It acts like a blood reservoir, especially during hemorrhage when there's excessive blood loss, during carbon monoxide poisoning, when somebody is exposed to excessive amount of carbon monoxide, during physical and emotional stress. So all these are points when large amount of blood cells are formed. They produce lymphocytes. Lymphocytes are one type of WBCs. In embryo, the spleen produces RBCs. It destroys the worn out RBCs. So with this, we finish off the circulatory system. So we have done the structure of heart, the various kinds of blood vessels. We have done the various kinds of body fluids and the various components of blood. So hope you have understood it all. Thank you very much and God bless you all.